The alternative pathway of complement activation does not need antigen-antibody complexes for its initiation. The alternative pathway of complement activation is a component of innate immune system. The alternative pathway of complement activation involves four serum proteins like C3, factor B, factor D, and properdin. Both gram-positive and gram-negative bacteria have cell wall constituents that can activate alternative pathway. Now, let us discuss about the steps of this pathway. The C3 contains an unstable thioester bond. The C3 undergoes slow spontaneous hydrolysis to produce C3b and C3a. In the next step, the C3b binds to the antigens of foreign cell surface or to host's own cells. In the next step, the factor B binds to the C3b present on the foreign cell surface and forms a complex. This complex is stabilized by Mg2+. In the next step, the factor D binds to the factor B in the previously said complex. Factor D then enzymatically cleaves the C3b bound factor B and releases a small fragment called as BA. Due to this factor D catalyzed reaction, a complex is formed called as C3b-BB. This C3b-BB complex has C3 convertase activity. The classical pathway of complement activation has a C4b-2A complex. This C4b-2A complex is analogous to the C3b-BB complex of alternative pathway of complement activation. When the C3b-BB complex is not bound to properdin, the half-life or T-half of C3 convertase activity is only 5 minutes. But when the C3b-BB complex binds to the properdin, the C3b-BB complex is stabilized. Due to this properdin binding, the half-life or T-half of C3 convertase activity of C3b-BB complex is increased to 30 minutes. The C3b-BB complex shows autocatalytic activity. By using this autocatalytic activity of C3b-BB complex, the C3 is hydrolyzed to form C3b and C3a. As a result, the initial steps are repeated and amplified. Some C3b binds to the C3b-BB complex and forms C3b-BB3b complex. The C3b-BB3b complex shows C5 convertase activity. The classical pathway of complement activation has a C4b2A3b complex. The C4b2A3b complex is analogous to C3b-BB3b complex of alternative pathway of complement activation. The non-enzymatic C3b component of C3b-BB3b complex binds C5. The BB component of C3b-BB3b complex catalyzes a biochemical reaction in which the bound C5 is hydrolyzed to form C5b and C5a. The C5b then forms the membrane attack complex or MAC. Please like, subscribe and share.